excited to talk to everybody about metabolic hormone signaling and physiology. Really, I'm going to focus my talk today on glucagon-like peptide 1 or GLP-1. I'll start with an overview of some key aspects of its physiology, and then we'll talk a little bit about GLP-1 pharmacology as well. So GLP-1 is a hormone that we make in our body. It's secreted from specialized cells within the gut, and it's secreted in response to eating a meal. And it acts throughout the body to elicit a variety of metabolic benefits. And of course, these benefits are why GLP-1 has become such a attractive therapeutic target for obesity and diabetes. So GLP-1 has actions on pancreatic islets to increase insulin secretion. It acts on the gut to slow gastric emptying and gastric motility. And I think that this is actually an important effect of GLP-1 that sometimes gets glossed over, important in terms of the pharmacology too. So this effect on the gut is part of the reason that GLP-1 receptor agonists are able to modulate or improve postprandial glycemia. It's also potentially linked to some of the adverse effects we see of GLP-1 receptor agonists. These can be associated with issues of the GI, like nausea, or even sometimes GI stasis, which is linked to kind of an overstimulation of this effect on the gut motility. GLP-1 acts on the brain, of course, to reduce appetite, and this is a key driver by which GLP-1 based drugs are producing weight loss. And then GLP-1 has cardioprotective effects and indirect effects to improve insulin sensitivity throughout the body. Perhaps one of GLP-1's most well-studied effects is its incretin effect, and this is something that's been of particular interest in my lab. So GLP-1 is an incretin hormone, which means that it potentiates glucose-stimulated insulin secretion from pancreatic islets. In other words, it stimulates insulin secretion in response to eating a meal. And this effect is really important. It accounts for up to 70% of our insulin secretion in response to feeding. There are two main incretin hormones. These are GLP-1, and the other one is GIP. It's another hormone produced by the gut that's also of interest pharmacologically. And this graph illustrates the incretin effect. So this is looking at healthy human subjects and their insulin secretion in response to glucose given through two different routes. So in the red line, we're looking at glucose being given IV, so bypassing the gut. And in the black line, we're looking at glucose being given orally. So this is the route where we're going to stimulate the secretion of those incretin hormones. And you can see that we get way more insulin secretion when we give the same amount of glucose orally versus IV. So the difference between these lines is the incretin effect. Another emerging concept in the field of GLP-1 physiology that I wanted to highlight, and one that my lab has done a fair amount of work on, is classically GLP-1's made in the gut, but can actually be made outside of the gut within the body as well. And one example of this is in the pancreatic alpha cell. So the alpha cell classically makes glucagon. Glucagon acts to increase blood glucose levels, but it turns out that there's a way to shift alpha cell hormone production to make GLP-1 instead. So essentially, there's a way to shift the alpha cell from promoting diabetes to treating diabetes. So we think this is another potential way to target GLP-1 in the future. So with that, I will shift to GLP-1 pharmacology. So overall, this is, I think, a really exciting time in the fields of obesity research, diabetes research, and nutrition research. Obesity is, of course, a difficult, devastating disease and a difficult disease to manage. And up until recently, we really haven't had a lot of pharmacological options to help folks manage obesity. And so with the development of GLP-1 receptor agonists, we now have pharmacologic options. So GLP-1 based drugs have become well established for the treatment of type 2 diabetes, obesity, and another concept illustrated in this graphic is that as we're seeing more and more people taking these drugs, we're realizing that there may be other disease applications as well. So we're seeing potential efficacy of GLP-1 based drugs on renal renal disease, liver disease, and even neurodegenerative diseases. Before diving in deeper into the pharmacology, I just wanted to talk briefly about the history of GLP-1, and thank you, Justin, for priming this. Just to kind of think a little bit about how we got to where we are today. So GLP-1 research, of course, has been going on for decades. It started in the early 80s with the identification of the sequence that encodes what would become known as GLP-1. In terms of milestones in pharmacology, I would say the first big one for GLP-1 was in 2005 when the first GLP-1 receptor agonist came on the market. This is exenditide or exendin 4. Oddly enough, it was discovered from the venom of the Gila monster. And it was a twice daily injectable formulation. The next big landmark in GLP-1 pharmacology that we just heard about was in 2014 when the first GLP-1 receptor agonist came on the market for the treatment of obesity. And this drug is liraglutide. It is a once daily injectable formulation. And then, of course, in more recent years, we've been seeing the development of even more effective GLP-1 receptor agonists and also the development of the multi-agonists that are proving to even be more effective than just GLP-1 receptor agonism on its own, which I'll talk about a little bit towards the end. So I wanted to just take a look at the data. So this is one of the really important clinical trials assessing the efficacy of semaglutide in the treatment of obesity. And you can see that it achieves upwards of 18% body weight loss here. Now, this field is, of course, rapidly developing, and one of the areas that folks have been working on is how can we make these injectable drugs orally available? And impressively, this has already been achieved. Semaglutide is orally available. It is on the market and approved for the treatment of diabetes. What current research is focusing on is whether orally available semaglutide can also be applied to obesity treatment. This recent clinical trial reports oral semaglutide achieves up to 13% body weight loss in human patients, which I think is pretty impressive for an orally available agent. And then lastly, I just wanted to highlight this concept, this new class of drugs that we're seeing, which are the multi-agonists. So the idea here is that, of course, we're seeing a lot of efficacy with the GLP-1 receptor agonists. How can we make them even better? Perhaps we can make them better by having them not just activate the GLP-1 receptor, but activate other metabolically beneficial receptors as well and achieve some synergism. And so, of course, there are a lot of potential combinations, and there are a lot of potential combinations out there being developed in various phases of development. And of course, we already have one that's on the market, tirzepatide, which is activating both the GLP-1 and GIP receptor, and at least in terms of weight loss, appears to be somewhat more effective than semaglutide. Another multi-agonist that's not on the market yet, but I wanted to highlight because I think it's interesting, is retatrutide. It's a tri-agonist. It activates the GLP-1 receptor, the GIP receptor, and the glucagon receptor. And this early clinical data, I think, look quite remarkable, showing upwards of 24% body weight loss in patients. So to wrap up, I wanted to end here because I think this kind of brings together why we're all here at this conference, which is to start to ask questions and think more deeply about how does nutrition fit into all of this. So I think there are a lot of relevant questions when we think about nutrition and GLP-1. One of them is, nutritionally, how do we best support individuals who are on GLP-1 based drugs? Even better, how do we nutritionally support people when they've come off of GLP-1 based drugs? And then another question that I look forward to us all discussing more is nutritionally, so independent of the pharmacology, nutritionally, how do we use the body's GLP-1 system and better augment that to achieve better metabolic outcomes? So I guess I will leave us with a bunch of questions and hand it off to the next speaker, who I think is Dr. Adams.